So where we left off was the four macromolecules, which are, what are they again? Carbohydrates. What's that? Nucleic acids. What was the tough one? What are the other two? Lipids and proteins. Good. Okay, so we're going to go through those one by one. I'm going to tell you a little bit more about them. I can find my spot. Here we go. So these macromolecules are what we call a polymer, and a polymer is basically a chain of repeating units. As we talk about these things, we're going to talk about so I would draw this graph on a separate piece of paper or somewhere off in the notes. We're talking about basically three different polymers. Lipids aren't exactly polymers, but they do have some structures similar to polymers. Okay? And all of them, though, in order to form bonds, it requires a hydrogen. Okay, and that's called, a water molecule, and that's called a dehydration synthesis. Okay? And then, so, when a bond is formed, you have an OH on one molecule and an H on the other molecule that combine to make a water molecule when that bond is formed. Okay? When a bond is broken, it requires a water molecule, again, to provide that H and OH, where there was previously just a bond. Okay? Those then are opposites, hydrolysis and dehydration synthesis. Okay, again, our four types of macromolecules essential for life are carbohydrates, nucleic acids, proteins, and lipids. And so we're going to start with carbohydrates. Move this over now. And carbohydrates have a 1 to 2 to 1 ratio of carbon, hydrogen, and oxygen, generally. So their monomer which is the single unit, is CH2O. Okay? Then we get into sugars. Okay, we'll talk about some other monomers. And the bond between those carbon molecules stores energy, has potential energy. And in plants, when you string these carbon molecules, hydrogen and oxygen molecules together, it makes starch. Okay, and in animals, we store these units of CH2O, in a molecule called glycogen. They're very similar, except they are structurally different. Okay, when we talk about sugars, we have monosaccharides. Okay, and these are 6-carbon sugars. There are other monosaccharides, but these are some of the most important ones: fructose, glucose, and galactose. And if you look over here, we talked about this briefly before, but fructose and glucose and galactose have the same molecular formula. It's all C6H12O6. But the double bond is in a different spot. in fructose and glucose. And so they are structural isomers. They're the same except the double bond is on a different carbon. Glucose and galactose are exactly the same except this OH and H are flipped. So they are stereoisomers. Okay, and they are a monosaccharide of more complex molecules, including disaccharides and polysaccharides. Okay, so you could put, let's just put glucose here. Okay, well, you could also put fructose and galactose. A disaccharide is made of two sugars. So lactose is an example that's found in milk. Okay, and it's made of two sugars. Sucrose is table sugar. Okay, and that's made of one glucose and one fructose. Okay, and then polysaccharides are three or more of these sugars bound together. In plants, they have cellulose and lignin, and that's what gives them the structural components in their cell walls, which makes them hard. Okay, the harder a plant is, or the stiffer it is, the more cellulose it has, or lignin it has. Chitin is another one that's found in fungus and also in insects, arthropods. Okay, and it is structurally can be very thick and strong. So the monomer for these saccharides, polysaccharides, are 6-carbon sugars. Glucose is one of those, but also fructose and galactose. Okay, nucleic acids, then, our monomer is a molecule called a nucleotide. Which has 3 components to it: a phosphate, a sugar, and a nitrogenous base. Okay, DNA and RNA are these are the most common nucleic acids, and probably the most important nucleic acids found in the body. There are some other ones we're going to talk about too. The difference between them, RNA has a sugar called ribose. Okay, that's why it's ribonucleic acid. And DNA has a sugar called deoxyribose. Okay, DNA also has one different base. So they have adenine, thymine, guanine, and cytosine. And then RNA has the same except it has uracil instead of thymine. Okay, so this here is a general picture of the 3 structural components of a nucleotide. You have your phosphate, your sugar, and your nitrogenous base. These are the nitrogenous bases, so this can be any one of these 4 in DNA or one of those 4 in RNA. The only difference between ribose and deoxyribose is that OH instead of an H. So it's an H in DNA, a hydrogen there. It's an oxygen and a hydrogen in RNA. These are bound together in a bond called a phosphodiester bond. Okay, and so that's what this is here. That bond is called an ester bond. You have a phosphorus in the middle, so it's phosphodiester. Okay, all of these then are nucleotides, and they're bound together. So DNA, again, it has those 4 base pairs: adenine, guanine, cytosine, and thymine. Well, for a lot of the class, just refers to them by their first letter. And the sequence of these is what stores information. Okay, so just like we use the alphabet to process and store information, they have a smaller alphabet, but it's the same thing. Different combinations will code for different proteins or different RNAs and can be passed from one generation to the other. DNA is arranged in a double helix, meaning it has basically two rows running in opposite directions of these strings of nucleotides. So this is one string here. These are the nucleotides. And then on the other side you have nucleotides. And they make bonds in between, which, if you remember, one of the important properties of water was hydrogen bonds. Okay, these hydrogen bonds are also formed between these antiparallel base pairs. And they're specific. So A only bonds with T or U in RNA, and G always pairs with C. You can see that here. T and an A, C and a G, T and an A. They don't match up right if you have like an A with a G. It won't work. They aren't compatible. Okay, RNA, there are lots of different types of RNA, but the three most important ones that we're going to talk about in this class are messenger RNA, abbreviated by a small m in front of RNA, ribosomal RNA with the small r, and transfer RNA with the small t. Okay, messenger RNA is a single strand of nucleotides bound together, and because it doesn't have another strand to bond with, it will bind with itself and won't really take any specific form. But it is very important for the process of transcription. We're going to go over transcription in a lot more detail later on. Ribosomal RNA is found in ribosomes. It's a very important structural component for ribosomes. Ribosomes help to make proteins. And then transfer RNA carries amino acids to the ribosome during translation. They're also important for making proteins. Okay? So just know these basics for now. Later we're going to go into a lot more detail as to how these work. There are a couple other nucleotides that are very important. This one, ATP, is the abbreviation for adenine triphosphate. That is used ubiquitously or throughout the body for energy. Okay, so that's the energy molecule in living things. All living things, even bacteria, use ATP. NADH and FADH2, you don't have to worry about what the long names are for those. But they are used as electron carriers in the electron transport chain, which is important for aerobic respiration, which is how we get energy from our food using oxygen. Again, just know that they are nucleotides for now. We are going to talk about them a lot more when we talk about respiration. Okay, our next one then is proteins. Another name for protein is polypeptide. So the monomer of proteins is amino acids. Okay, but proteins then are what does the work in the cells. They are catalysts, so they are involved in enzyme catalysis, which means they help reactions, they help break things down, This is hydrogen peroxide, which is a destructive organism or molecule. If you don't break it down into water and oxygen, it will start destroying the cell. So we have the enzyme catalase to break that down. They're important for defense, so including antibodies to help fight microorganisms, viruses, bacteria. They're important for transport, so they are embedded within the walls or the membranes of cells to allow things in and out of the cell. They're involved in support, including muscles are made out of protein. Motion is also, we require muscles for those. And they also help regulate other processes, again, through letting things in and out of the cell. And they also will store, there is energy stored in the bonds of protein. All right, again, we have amino acids, and they're bound together by peptide bonds. So if you wanted to do another layer to your graph here and put bonds, we talked about the bonds between nucleic or nucleotides are phosphodiester bonds. Okay, the bonds between amino acids are called peptide bonds. Okay, which is why it's also called a polypeptide. Poly means many, so you have many of these peptide bonds. Okay, the structure of an amino acid, at the center of it is a carbon atom. Okay, what did we learn about carbon atoms last time? What is unique about carbon atoms? You can look in your notes if you want to. But this is important. This is a very important part of why carbon is found in is the most abundant molecule in living things. Make covalent bonds with 4 different elements. Okay. So we have a central carbon. Guess how many things are bound to it? 4. Okay. You have a hydrogen by itself. Okay. On every amino acid, it has one of the bonds to the central carbon just attached to a hydrogen. Okay. You have a carboxyl group, which is this COOH, okay, molecule. And then you have an amino group which has nitrogen. And then you have an R group. And the R group is the variable part. So you can have all sorts of different structures. But they are within 20 different arrangements of amino acids. Okay? The peptide bond is formed between the carboxyl group of one and the amino group of another by a dehydration synthesis. Okay, so when they come together, you lose the water, or you gain a water. All right, so you don't have to know all these. This is just to illustrate the different R groups and the different names of our different amino acids. Some of them are polar, some of them are non-polar. What does polar mean? Remember that from last time?
One molecule has a partial charge and the other molecule has the opposite partial charge.
Right, so there's a charge to it. They do not share the electrons evenly, okay, which makes that charge. Non-polar bond is an evenly shared electrons between the molecules. So these are non-polar, these are polar. You have charge, so they have a negative charge on it or a positive charge on it, an ion at the end, basically. There's a few called aromatic. And they call it that because they actually give off a little smell. But that also refers to this ring structure. Okay? All these different structures give different properties to different amino acids. And you string them together in different arrangements, and that gives the properties of your different proteins. Okay, and that's basically what we're going to talk about now. So there are 4 different structural levels which give the overall protein its function. Okay, the first is the primary structure. So that's just looking at the sequence of amino acids. So you may have a valine, a proline, an isoleucine. That structure, so just like, this would be like the letters, okay? If we're comparing it to the English language, this would be our alphabet. The secondary structure then is the interaction between the groups of the R groups within the chain. So this would be like words of a sentence. So this would be like letters of a word, words of a sentence. Okay, and you can shuffle around those words and make different sentences. So this figure is showing you already have the sequence set, but then you have interactions between or among the chain. That's the secondary structure. Okay, one of these is called an alpha helix. Another one's called a beta sheet. An alpha helix would be this kind of curving structure there. And then a beta-plated sheet is what I pointed to before. You have these sheets of molecules that are interacting, forming bonds between each other. The tertiary structure then is the final folded shape, and it includes all the secondary structures within there. Okay, so this would be like sentences in a paragraph. Okay, and then the quaternary structure would be if you have multiple polypeptides interacting among each other. So the example of that is hemoglobin. We talked about that before. It has 4 globin proteins which interact. This is illustrating that. All of these globins have the same structure, but they also make bonds between each other. Some proteins are just made up of one tertiary structure, so they don't really have a quaternary structure. In addition, in order for proteins to gain their function, do you guys need me to go back? Okay. Just yell at me when I do that, when I go too fast. Good? Okay, so in addition to the sequence of amino acids giving the proteins their structure, they also needed to be folded in a specific way. So even in order to make those, turn off my phone. Yep. So in order to make those different secondary structures, and in order to make those R groups interact, you have what are called chaperonins, which just take the amino acids, the polypeptides, and folds them in the correct shape. Okay, so this one has a little hole within it that the protein goes into. It has some sort of reaction or probably has enzymes in there to fold it correctly and some sort of top on it, and then it lets it out. If a protein is exposed to too much heat or too much cold or too much too many ions, too salty conditions, it may denature, which would, the protein itself may not change, but the actual folding of the protein may come undone. Okay? And that's called denaturation when it becomes unfolded. It loses its shape, loses its function. All right, our last one then is lipids. Okay, I'm going to put a star next to lipids because they aren't really a polymer. Okay, but they are made up of some specific pieces. Okay, they are defined as nonpolar hydrophobic molecules. So what does that mean when you mix them with water, or when you try and mix them with water? Yeah, they don't. They don't mix with water. That's what defines them as a lipid. So this includes fats, waxes, oils, and then there's also some lipid vitamins. Some hormones are lipids. For example, this is testosterone. This string structure here is a nonpolar structure, so it doesn't combine with water very well. This is some earwax. Okay, that's really the only place where we produce wax. Here's some olive oil, and this is some bacon grease. Okay. So lipids then are made of one type of lipid is fats. Okay, and they are made of let's put that actually over here. Okay, they are made of a glycerol backbone and fatty acids. Okay, and you can have a triglyceride, which will have 3 fatty acids on one glycerol, or you can have two diglyceride or a monoglyceride, which would have 1 or 2. But generally, if there's enough energy and enough fatty acids available, they will make a triglyceride. Okay, we can separate those into 2 categories: unsaturated and saturated fats. And the difference between them is sometimes they will make these double bonds between the carbon molecules. And what that does is it makes a kink in the chains. Okay, these are fatty acids. If they don't have any double bonds, they will just be nice and straight. That allows those 3 fatty acids connected to the glycerol. And this is the glycerol on the side here. That allows them to compact together really well. Okay, that means that it's going to require more energy to break them apart. Okay, so at room temperature, they will be solid. Okay, most animal fats are going to be saturated. Okay, an unsaturated fat are going to have these double bonds, so it's going to be kinked, so these fatty acids can't bind together. It's not going to have require as much energy to break them down a little bit, and so those are unsaturated fats, and those are liquid at room temperature. So those are going to be mostly your vegetable oils and other oils. Okay, these are not very good for you if you eat too many. These seem to be a lot better for you as far as eating. Okay, another couple types of lipids. We have phospholipids, which are structurally different than our fats. They have a phosphate head, which is actually hydrophilic. Okay, so it will make hydrogen bonds with water. And then you have a glycerol, which is basically just a connector. And then you have 2 fatty acids. Okay, and these are important for making if you have all of the phospholipid heads pointed towards the outside, and the lipid tails pointed towards each other, you can make a phospholipid bilayer, and that's what makes up your cell membrane. Okay, this is important for every cell. They have to be compartmentalized somehow. The phospholipid bilayer is how that works. Okay, another example of where these are used is in micelles. Micelles act kind of like detergents in your laundry or in your dishes. They have this phospholipid shape, so they are part hydrophobic and part hydrophilic. And so to get the grease off of like out of a pan, you put in a detergent, you use soap. What happens is the lipid portion will bind to the fat. The phosphate portion will bind to the water, and then they will all go out together. Okay? Same thing happens when you wash your clothes, get all the oils out of your clothes. Okay, these are not used in your clothes, though. These are used in your stomach to help break apart the fats that you eat. So it physically helps break globs of fats down into smaller pieces so they can then be digested and absorbed. Okay, that's it as far as our lecture goes. This should help you keep everything straight. At least this would be a good place to start. You do need to know the other details on there. And then the rest, so you have the rest of the time to finish or work on the crossword, and then turn this in, and then you're free to go.